The overall goal of these multi-well-based sequence assays is to better understand the effects of cold physical plasma over oxidants to biological responses in vitro using optical emission spectroscopy, liquid analysis, and cellular activity, microscopy, and flow cytometry assays.
This method can help to answer key questions in the field of plasma medicine, especially with regard to plasma-derived reactive species important for the induction of immunogenic cancer cell death. The main advantage of this technique is its semi-automatized approach. It uses 96 well plates to increase speed and productivity of the research.
The implications of this technique extend towards the future plasma therapies, like in dermatology or oncology, and as they provide the means to identify the plasma-derived reactive species which are relevant for the biological responses.
Demonstrating the procedure will be Felix Niessner, a technician from our laboratory.
For plasma species monitoring, position an atmospheric plasma jet in front of an optical emission spectrophotometer perpendicular to the plume axis, and use the dedicated software to record the photo emission and wavelength of each gas condition at 2 standard liters per minute feed gas flux. For plasma-treated superoxide analysis, set up a final protocol for the XYZ table with the appropriate respective treatment times and well positions. Then, add 100 microliters of freshly prepared master mix to the wells of a clear, flat-bottom 96 well plate in triplicate, and use a microplate reader to measure the absorbance at 550 nanometers.